And for those who enjoy this channel and would love to support us financially, please feel free to hit that donate link. We'd greatly appreciate it. God bless.
Looks like we are live tonight on Standing for Truth. Better late than ever. It's nice to see everybody tonight. We've got a packed house. This is going to be a lot of fun. I'm looking forward to it. So a couple brief announcements first. We've got a good week ahead of us. Let me see here. We've got this Thursday, Dr. Dino himself will be back debating Derek Barnes on Is Evolution Science? You'd remember Derek, I debated him on Modern Day Debate. Nice guy. It's going to be a really good debate. He's excited. Dr. Dino's excited, so that's going to be a lot of fun. Also, Dr. Stadler, I'm going through his 2 books here, and he should be on probably next week sometime. We are going to be discussing his newest books, must-reads. Lots of good stuff there, so looking forward to that one. Right here, just got my new Devolution shirt in. We've got the Pond Scum to People shirt, mug, all our books going on, guys. So make sure you're checking out the webpage if you want to be rocking all the SFT gear. So tonight, I should say, I hope everybody is well in the chat tonight. We are going to be addressing some arguments from the critics on created heterozygosity, as well as a few other things. Originally, I was going to go through a website, a blog from some critics of replacing Darwin and Dr. Jeanson. We'll try and get to that at the end. But first, we're going to try and cover a few other things before we get to that. So let's check on the chat here. It's going to be a little interactive today. I'm going to be screen sharing a bunch of things. Get some water here. Okay, I'm going to be doing some screen sharing, try and make this visual. Some of this stuff is going to be engaging. Good to see everybody in the chat. Okay, let's see here. Actually, one thing before we get started that got me kind of laughing here is we've got them backed in a corner. You can see on the different YouTube channels of our favorite critics, you've got a lot of these evolutionists. I mean, you got this one here, which is funny. You'll never get the militant ones, right? You're never going to get Dan. You're never going to get Gutsick Gibbon admitting that we're presenting arguments that are difficult for them to address. That's why we're referring to them tonight as Team Dodgeball. Here we've got good old Stacy, who's saying some creationists make decent arguments if we are not honest with ourselves. But she's looking for more debates and admits down here, I get a kick out of these comments sometimes because I always say it's about the ones that are on the fence, the ones that are agnostic, the listeners. You know, they're the ones that are seeing that. As you can see here, she says, but honestly, and that's what we like. We like honestly. They appear to be winning the debates. And she's calling out for Rational Mind, Evograd. He's a keyboard warrior. But, you know, a few of them to kind of step up their game. So I just kind of thought that was funny because I see this all the time. Our last few videos on Gutsick Gibbon, you know, I've seen some comments asking for her to respond because their worldview is crashing before them, you know, but if only they would just admit the truth, convert, then they wouldn't have to look to their apostles of the science fiction religion of evolutionism to, you know, defend indefensible position is what it is. So like I said, we're going to be touching on some technical stuff here. So why don't we start with, I've got some notes just from some arguments that I've seen. I'm looking at the chat now. Good to see everybody. But like I said, unfortunately, I'm going to have to be screen sharing soon. But I will be looking periodically in the chat. Good to see 30 people here. We've got some Let's see. We've got some super chats already. Thank you so much. And some super stickers. Catherine Stormguard, you're famous. Yeah. Well, and like I said, the other night we did a few-hour video on Vice Rhino, and we challenged him to refute the video. But we know these videos are long. There's a lot for them to touch on that they, like our mutation rate videos, incredibly detailed, I would say irrefutable for the most part. Therefore, anybody that we refute in a nightly video on our new series, another evolutionist bites the dust, we are also challenging to debate. So we've got Vice Rhino, Professor Dave, Pelagia, Aaron Ra, Professor Stick, all of these guys, we've got a lot of fun in store for them. So, I'm looking forward to finding out if they'll take up the challenge. Our other challenge was we got brother Anthony Maurice in the chat. We had Dr. Ryan Hayes on last week. We demolished abiogenesis. And these critics, they like to scoff at, front-loaded genetic diversity, an initial creation event that would explain the biodiversity that we see. And we proposed a challenge to these critics of biblical creation to respond to the interview that we did with Dr. Hayes. The last few videos I've done, I broke it down for about 10, 15 minutes. Just a bunch of questions that I want answered in regards to origin of life, abiogenesis, there's chicken and egg problems after chicken and egg problems, chirality problem. I'm not going to reiterate it here because I've already done it a few times. I imagine the critics have seen the videos, they've seen the interview. So man up and take the challenge. Let's see what you got. Okay, so let's see here. What I'll do here is I'll start off by sharing screen on a paper that I'm not too sure the evolutionists are, as long as you can see the screen. Okay, let's go. So this is a paper that you'd have to download. I've got it in front of me. Throat's a little dry. Just finished a workout and rushed into here. So excuse me if I'm taking some drinks. Also fighting a cold, so voice might be a little scratchy. Forgive me. So this is a paper you'll want to download. I've actually got it in front of me. I've looked it over a few times, studied it intensely. Adam and Eve, design diversity and allele frequencies. So one of the major arguments from our critics here, Dr. Dan, for example, Dr. Joshua Swamidas, he's notorious for it as well. What's funny, though, when this paper came out, this was a newer paper written by Dr. Sanford, Dr. Carter, Dr. Baumgartner, so some solid authors on this paper, some really, really good numbers, numerical simulations, and that's what they're always asking for, right? The math, the numbers. Well, here, and I can assure you, here in the abstract it says, and it's a very technical paper. I just want people to know that it's out there. Look into it. I haven't seen any criticisms or rebuttals from the critics on it. One funny thing about it, unless Dr. Swamidas has changed his mind, on his website, Peaceful Discourse, when this paper first came out, he admitted that this seems to give a plausible explanation for the allele frequencies in people groups today. In regards to all coming from Adam and Eve. And he said that, okay, they've got an explanation for this, but they have a problem known as the TMR4A problem. It's a sophisticated population genetics argument. So I'm going to deal with that one after I deal with this. So even he's admitting that the numerical simulations done here in this technical paper does seem to give validity to all human beings today descending from two people, Adam and Eve. So it says, in this paper, we have critically examined these arguments. Our analyses highlight several genetic mechanisms that can help reconcile a literal Adam and Eve with human allele frequency distribution seen today. We used numerical simulation to show that two people, if they contain designed alleles, can in fact give rise to allele frequency distributions of the very same type as are now seen in modern man. I challenge our critics. Gutsick Gibbon, if she'd like. Dan, pull up this paper. Like I said, it is a lengthy paper. Go through it, refute it. In the conclusion, you won't be able to see it here, but I've got it in front of me, so I urge you to download it. I typically download these and print them so I can study them, highlight it, for example. So let's see here. So in the conclusion, they say, in this paper, we have used logic and numerical simulation to show that there are several design diversity mechanisms that can reconcile a literal Adam and Eve with the allele frequency distribution now seen in the human population. These genetic mechanisms include design diversity within Adam and Eve's 4 sets of chromosomes, followed by accelerated genetic drift associated with multiple population constrictions. As above, combined with more powerful demographic forces such as selective sweeps, lineage extinctions, and differential subpopulation expansions. And thirdly, design diversity within Adam and Eve's original created genomes. So you can go through this, very detailed, very technical. Like I said, unless he's changed his mind, Dr. Swami Das, a militant young earth creationist. So you can look into it. Like I said, he might have changed his mind. I don't want to put words in his mouth, but the last time I looked, he seemed happy with this paper. It's a newer paper. And he brought up another argument, the TMR4A. And I, the other day, refuted that one. It's a sophisticated population. Tonight's going to be kind of technical, but it's mainly because Dr. Dan, Damage Control Dan, he put out a video that was kind of technical as well. So I'll kind of give the basic rundowns on how to refute these arguments like I'm doing here. I'm showing you some key sections in it, but if you want to go down deep, stay down long, come up dry, download the paper, read it all for yourself. They're always accusing the creation of not doing the math, not doing the numerical simulations. It's been done, okay, ad nauseum. And the presentation I'm going to give after that, having to do with what's called the HapMap data, you're going to see that for yourself. So just wanted to start off with this one. But I wanted to start off with the HapMap data. But you know what? Since I'm already on this issue with the TMR4A, okay, Dan in his video, I linked it. He is really relying on this, and he got it from Joshua Swamidas. And I pointed out in a stream the other day that it's based on a circular argument. Okay, it assumes away created heterozygosity. Now, in his video, he says to the contrary, it assumes created heterozygosity. Well, what I want to point out is that it's kind of, here's the problem. So Dan got it from somebody who has put forth an erroneous argument. So now Dan is now putting for, it's like a ripple effect, erroneous argument after erroneous argument being parroted. It's kind of like Gutsick Gibbon, where she recently got caught copying Swami Das and Evo Grad, for example. I asked her several questions on the Y chromosome papers. She provided 2 sources. We'll see if she answers those questions, but she's also always parroting the whole triad versus dyad argument that supposedly Dr. Jeanson is looking at somatic mutations. He's looking at dyads, should be looking at triads. Well, I showed in a stream a couple nights ago that dyads and triads have been done in autosomes, and they both arrive at similar results. Actually, I think I have that here. Let's see, this is right here. Unfortunately, the critics are kind of out of date on things. So estimating the human mutation rate from autozygous segments reveals population differences in human mutational processes. I'm going to look for my phone real quick because I want to make sure that the screen is working well. Like I said, usually I'm interactive with the chat, but tonight I'm going to be showing a lot of slides. So I'll look in periodically. Super chat from George, $20. Thank you so much, George. Get some lemon and honey added to a hot cup of tea. It will fix your sore throat. That a boy looking out for me. I think I will do that actually after this. If I start losing the voice midway through, we may just make this a 2-parter. So, okay, good to see 40 people in the chat. So have a look at this paper. I'd like to see Erica address this one as well, the questions that I had for the Y chromosome. Okay, so let's go back to, like I said, this paper, have a look at it. Okay, so this whole TMR4A argument, like I said, a lot of this is technical, but let's deal with this right away because we're gonna slam this one. We're gonna put this one to bed for good. And what's funny is, from what I'm hearing, there's been no rebuttal to this paper. But supposedly, as good as this paper is, we can't explain this TMR4A problem. So, but here's the problem with that, okay? This TMR4A argument, it's a circular argument as it pretends, okay? It pretends, and I want to emphasize that, to assume created heterozygosity, when in fact, TMR4A has heterozygosity as part of the analysis, but it is nothing like the model of created heterozygosity. Time to most recent 4 ancestors, okay, or 4 alleles, is what this means. And I suspect that there's not too many critics looking into it other than Swamidas, and therefore they themselves haven't looked into it too thoroughly. Well, what it does is it traces the origin of our autosomal DNA differences back in time, okay? Incorporating recombination and mutation. It allows the researcher to follow lineages, and this TRM4A point is when 4 lineages are present in the human population. This is essentially, okay, if you're following me, this is essentially the point where 2 lineages could be present in Adam in a heterozygous state, okay? In all, for anybody new here, created heterozygosity just means that according to our model, God would have front-loaded Adam and Eve with pre-existing functional DNA differences, pre-existing genetic diversity. It'd make no sense for God to create Adam and Eve genetically homogeneous, because when he said to be fruitful and multiply, would he want that to be carried out through cloning? Of course not. That wouldn't make any sense. So back to the point here. You would, as I said, these 2 lineages could be present in Adam and 2 in Eve, okay, both in a heterozygous state. But the entire analysis still treats every difference as the result of mutation. Okay, let me say that again. But the entire analysis still treats every difference as the result of mutation. Here's the problem. This is why it pretends to assume created heterozygosity. We assume the vast majority of DNA differences, nuclear DNA differences, are the result of initial created heterozygosity. And this is why your critics, they want to ignore your uniparentally inherited DNA, your non-recombining DNA, like the Y chromosome, the mitochondrial DNA, where there's just so few mutations. And the mutation rates are fast, and they only take us back max 6 to 6,500 years. And those DNA compartments, we agree with the evolutionists, they are the result of mutation. That's why the non-recombining DNA, like the mitochondrial DNA, the Y chromosome, this would be the best place for us to test models in terms of origins, in terms of ancestry. But they want to obfuscate the issue, hide the fact that they're losing in the nuclear DNA, where things get a little bit messy. We inherit 2 copies, much, much larger, of course, and we assume the initial created heterozygous state. Now, here's the thing. As I pointed out, the entire analysis still treats every difference as the result of mutations. It just traces their mutational heritage in a more sophisticated way, as I was talking about earlier. This comes down to kind of complicated population genetics. But in contrast, the created heterozygosity hypothesis treats, as I said, millions of DNA variants as the result of de novo origin, de novo creation. In a sense, in the terms used in this TRM4A analysis that Swamidas likes to talk about and says hasn't been solved, the initial created heterozygosity, the model itself, puts millions of lineages in Adam and millions in Eve. So this is where it comes down to the straw man. This is why they pretend that it assumes created heterozygosity when in fact it doesn't. And all that to say that, yeah, it's a little bit technical and a little more detailed than is necessary. But considering these are the arguments that they're putting forth, we're just going to have to deal with it. So once again, I recommend read this paper. The critics have a problem with allele frequencies. It's been dealt with. The numbers have been looked at. Numerical simulations have been done. Let me go to the slides that I put up here. Okay. Actually, here, we'll get to this after. Dan is dealing with what's called hypervariable regions in the genome, where there are more than 2 alleles present. You can get up to hundreds, in the thousands. We're going to deal with that after. So I've got some slides here. Now, I took this from a lecture from Dr. Rob Carter that, once again, I don't believe the critics, very few of them have looked at, studied. This breaks down the numbers, breaks down what they're looking for. So Dr. Carter and also Dr. Sanford, who you'd be familiar with, looked at a program. If you're familiar with it, it's called Mendel's Accountant. Okay, it's actually the most sophisticated evolutionary modeling program ever written. And guess what? It was written by creationists. But what was it done? Why was it done? It was done to model the evolutionary story with real numbers. And it's funny because these critics, as I said earlier, they're always asking for the numbers, and yet have clearly never read or examined these large numbers of papers. I just showed one, and articles going over the exact numbers. I invite them to check it out for themselves. Or maybe they'd rather just pretend they don't exist, because that's oftentimes what they do. That's what Erica did with Dr. Jeanson's Y chromosome, his second paper that he just released, where it goes over the testable predictions. You know, why are they working so well if there's so many omissions, apparently? Now, here's the thing. In the videos and papers, articles, of course, on this topic, written by PhD creationists, you'll find models like this, okay? This specific one here has to do with some of the typical results you get for a common model run using a human-like population over long periods of time. Now, Dr. Carter, in the video where I took these slides from, it's called the non-mythical Adam and Eve. It's really technical. I've had to watch it a few times, 3 or 4 times, study it, take notes. Really, really good. Must watch. I'll link it in the video as well, or in the description box. For sake of time, I'm not going to go through all the details. The details can be seen in that video and in these papers and articles that I'm referring to. That's just the nature of the numerical simulations, the numbers, the mathematics. You know, I know it can be a little boring for an audience, but it's necessary given the rebuttals and videos and criticisms that are coming out. So Dr. Carter, like I said, in this video that you'll watch, I got some slides here, give you guys some visuals, explains that these are mutations that have occurred in the population since the run would have started. You can see that most of the mutations are rare, okay? If you're looking at, this is what I'm referring to here, Mendel's Accountant model run. You can see that most of them are rare because they appear in a single person. But also, most of these rare mutations will drift out of existence. This is just how it works. We know that most mutations are lost. Nobody's going to disagree with this. It takes a long time and a lot of work for random genetic drift to get mutations to reach a high frequency. Okay, now here's the thing, though. The critics will complain. We're cherry picking the numbers, cherry picking the data to fit our preconceived idea. Well, there are many different parameter settings that reach the same result. Dr. Carter points this out actually in his articles, papers, and in this video. And like I said, the video is called The Non-Mythical Adam and Eve. And he points out actually that what we see in these simulations are not consistent with the HapMap data. Actually, I should, before we get started, just to not confuse anybody, you've heard me say it before, HapMap. I talk a lot about it in my new Refuting the Critics book. What is it? Okay. Well, HAP stands for haplotype, and, well, map, I'm sure everybody's familiar with what a map is. You'll see Dr. Carter explains in the video that I highly recommend watching. I challenge the critics to go through it, refute it if you got a problem with the model, the data, and the evidence. So he explains what I'm referencing right now, but a little bit in more detail. Let's break down what a haplotype is and what a genotype is for you guys. A genotype is a string of nucleotides string of DNA. Now, most of us are actually invariable. And so what is most important to sequence, of course, would be that which is variable, meaning the variable places in the what? In the genome. For the HapMap, what they did was they actually sequenced over 1 million letters. And so a haplotype would be a series of SNPs, okay? And this is what we're going to be looking at here. So let me see if I can move slides here. Okay, so we're going to get into Adam and Eve. Okay, so now that we went over what HapMap is, the HapMap data, what they did, Dr. Carter points out that what we see in these simulations, contrary to what the evolutionists might want to admit to, is not consistent with the HapMap data. What evolution says in the HapMap data, they're in contradiction. Now you can see a straight line. Okay, so over here, you can see a straight line where there are many variations at the 40 and 50% level. We would not expect this given the evolutionary numbers. Dr. Carter explains all this right around the 20-minute mark. So like I said, I'm going to link that information in the description, because if I were to go through the entire video, all the details, we'd be here for 20 hours. Not that we have never done a 20-hour video. Like I said, it's a must-watch lecture. I challenge the critics to debunk what I said on the what I was talking about earlier with Swamidas, and also I challenge them to refute the paper on allele frequencies as well as this video. The math has been done. The numbers have been done. Like I said, Mendel's accountant is the most sophisticated evolutionary modeling program there is. So like I said, I've personally watched this lecture many, many times, and it's going to take some study, and it's definitely going to take some assessment by the critics. So let's see, we're going to go into, okay, this is kind of where it gets interesting and technical. So anybody has any questions, email me, of course. I'll be happy to help. And also good news, I think we're going to have Dr. Carter on hopefully within the next week, which is awesome. He would be the #1 pick for me to interview. And I've been talking to Paul Price, and Paul Price, I think, is going to get him on for us. So he'll, hopefully we got any really technical questions, he'll be here to help us. So actually this all worked out nicely, because we'll be going through this run before he comes on and helps us out. Really looking forward to it. So like I said, so we're looking at Adam here. And Carter, once again, he wrote a program, you can see him here in the slide, where he modeled human populations, okay? And this is what they say we haven't done. This is what they say doesn't exist. Like, it shows how out of date they are. And you'll see that more and more as this goes on. So he started with 1,000 variations in Adam, okay? Adam having 2 of each chromosome. If one chromosome has an A, the other one would have a G. Adam as a population, the first man, of course, will be treated as 50-50, okay? To make things simple, Carter assumes Eve had all the same variations. We assume she's a clone. There's good reason to say that she wasn't, which would add even more genetic diversity, but it's not necessary. So we'll assume the clone approach. But she'd still have DNA differences within herself, of course. Now, if we take 2 heterozygous individuals and you breed them, what you're going to get 25% of the children are going to be AA, okay, capital A, and 50% will be AG, 25% will be GG. You can see here, you learn this in biology in high school, the old Punnett square. Okay, so every single one of Adam and Eve's children, as you can see here, another graph. I know this is all kind of technical, but the visuals are nice. Like I said, I've had to study it a few times. I do want to point out, I know that genes are inherited in chunks, and this is all dealt with. For now, we're assuming no linkage, okay? And I'm going to make a prediction that a lot of the arguments I receive on this video are directly addressed in the papers, articles that I refer to, as well as the lecture itself. So we'll see if that testable prediction is fulfilled. Yeah, so based on what we talked about earlier with heterozygous individuals, every single child of Adam and Eve would be drastically different, okay, in one generation. And you can even see this because above here, straight line, we can see that it's changing all in one generation. And what's funny is Dr. Carter talks about how he's pooled all the data and gone into incredible detail showing how consistent the genetics and the math is with Adam and Eve. This is actually really fascinating stuff. So we get to see the results of 4 children over here. We get to see the results of 16 children and so on and so forth. But regardless of the matter, when you go from Adam and Eve to the next generation, you get a tilting of the allele frequency spectrum, okay? And you can see this in the slides. This is all one step in the right direction, but let's continue. Also, like I said, please, because I know this stuff's technical, might even be a little bit boring, but it's necessary given the arguments that have been put forth to us. So read the papers, watch the lectures, and also gather some questions for Carter. Hopefully next week we can get him on. Carter also, I want to point out too, leave no stone unturned. He does account for the, what he does here is you can see it down here in the slide. He accounts for invariable places and mutations. For this slide, he added 100 alleles that were invariant in Adam, okay? So this line right here is where we are heading towards. Genetic drift and mutations over time, and we are beginning to make our way to what we see today. Exactly what the evolutionist says we can't account for, which is what I find funny. And also in the HapMap data, okay? We'll talk a little bit more about that. Now, Carter takes no corners. Probably knows he's going to get criticism. Takes no corners. As you can see here, he even uses overlapping generations, which is fascinating considering how, because a lot of models are more simple, basic, and they don't do this, but he considers overlapping generations, considering how many kids the early patriarchs were having, and also their long ages. Like I said, I invite everybody, especially the critics, to look into all of this for themselves. The data is out there. The modeling has been done. The critics are out of touch with reality. Now, based on the parameters in the modeling, okay, after 100 years post-creation, the population is going to look a little more different. Now, eventually, after hundreds of years, okay, in the exponentially growing population, what happens? Well, genetic drift stopped. Once the population got above a couple hundred people, we can see that the next generations get larger and larger, where Dr. Carty actually explains that they are grabbing all of the genetic variation the previous generation had, and they add it into the next generation, okay? How many people in the world today? Well, we've got about 7 billion, maybe a little bit more. What does this mean? Okay, well, genetic drift isn't operating, or at whatever level it is operating, it's at a very, very slow, very low levels, okay? Now, at this point, we are still not quite to what we see today, but what comes next in the biblical model? Anybody in the audience can guess? Of course, the flood. And what happened at the flood? 3 full sibling brothers and their wives, whom form a new population that is called what? Inbreeding. And we know that inbreeding leads to what? Genetic drift. And that's what's important here. So eventually we see exactly what we see in genetics today, and more specifically in the HapMap data. Small population followed by rapid and exponential growth, the flood bottleneck, rapid genetic drift, and of course, rapid mutation fixation, faster substitution rates in this case. It's all been accounted for. It's all consistent. The numbers are there. The model is very, very consistent, even more consistent in the biblical model than the evolutionary model, which is funny because they're the ones saying that we don't have any of the numbers, any of the data. So yeah, just covering this briefly was kind of technical, I know. Make sure to go watch the entire lecture. Also read the linked articles and papers. It's really, really fascinating. So email me, of course, or save some questions for Dr. Carter himself. And like I said, I predict that the objections we get will be addressed directly in these videos, papers. And they complain that sometimes we do 3-hour videos. So we could go through it line by line, word by word. But like I said, we'll be here for 10 hours. So now if you go here, since we're talking about the HapMap data, this is from my book on pages 27 to 30. Now, I love the results from the HapMap project, okay? And I've been talking about this for a long time, and I never get any answers. So here's the thing. Like I said earlier, this is right from my book, Refuting the Critics, pages 27 to 30. The HapMap project was designed to catalog a significant fraction of human genetic diversity. What they did was they analyzed millions of variants in thousands of people from around the world, like I was talking about earlier. And they made this data freely available. One might wonder, and this is actually Dr. Carter again, I'm quoting, if the HapMap is so unfriendly to the creation model, why do I, being Carter, have much of that data stored on my hard drive? In fact, I am quite happy with what we have learned from the project. He says, I am even happier with the 1,000 Genomes project, which is a step above HapMap, but it's not complete. So what did we learn? Here's a couple things. I talk about this frequently. Now we're just going to look at it in Carter's words himself. The human genome is young. Shared blocks of DNA are large, and there has not been enough time to scramble them to randomness. Okay, let's think about this real quick. We know crossing over takes place, gene conversion, recombination. If we've been evolving for millions and millions of years, from the Australopithecines to Homo erectus, we've had an out of Africa event, eventually to Homo sapiens, there's been a lot of genetic mutations accumulating, of course. The evolutionist explains the origin of all genetic diversity as a result of mutations over time. There's many problems in that to begin with, but besides the point, there would be a lot of recombination, there'd be a lot of shuffling, gene conversion, crossing over going on. In hundreds of thousands of years, okay, since their origin of humanity, the genome, there wouldn't be any of these shared blocks of DNA that are large. These large linkage blocks that exist in our genome, they should be scrambled to nothingness, okay, in deep time evolution. But because there are so many large linkage blocks still, it's as if there's certain parts of the genome that haven't even been shuffled. They haven't even experienced recombination. That speaks of the youthfulness of the genome. And I've been asking that question over and over again to the critics. Erica says she's finally going to respond. And Dan did, recombinational hotspots, cold spots, which is a thing, but it's also a circular argument because they know that the genome can't be young. Therefore, they want to look to that which can't really explain why there are so many large linkage blocks. It's not going to solve the problem, is the point. Secondly, the human population came from a single source. Most blocks are shared among all world populations. Thirdly, I like this one. The human genome is falling apart. Deletions tend to not be shared among populations, but are unique to subpopulations. This is further evidence for the youth of the genome and that we came from a single source population in the recent past. Okay, let's see here. We're going to get into, I told you a lot of this would be technical real quick. Let me go back to anybody in the chat. Good to see everybody's doing good. Super sticker from Doki Doki. Thank you so much. Tony, my man. How was the debate tonight? Shadow Dancer, thanks so much for the super sticker. Yeah, definitely watch the HapMap presentation by Carter. The first time I watched it, it was like, okay, I'm going to have to watch this a couple times. But I just want to point out the fact that the numbers are there. The simulations have been done. They, Adam and Eve, design diversity and allele frequencies. I mean, definitely check out that paper. So his next big argument, though, is hypervariable positions. Okay. Now, first off, I want to point out, and he doesn't see, Dr. Dan, I should say, doesn't seem to point this out. He talks about it a lot in his most recent video, which I've linked. Definitely watch it before you watch this. Now, it's a fact that some regions of the genome actually seem to be more prone to mutations than others. Okay. Because he's looking at the exception to the rule where we've got hundreds to sometimes thousands of alleles. How do we account for those, right? But the critics still underestimate the amount of allelic diversity that truly exists, especially when here's the biggest issues. When defining alleles as versions of a gene, then a large number of alleles can arise from created heterozygosity. Are they defining alleles as single nucleotides, meaning the maximum of 4? So you have to define the allele correctly. Let's see. SFB mob. Good stuff using the evolutionary models to confirm genesis. I love to do it, brother. I love to do it. We've Shadow Dancer. When do evolutionists think we became biologically human? So I guess Homo sapiens sapiens, 200,000 years or 100,000 years or recent past. Well, the numbers fluctuate. Their Y chromosome numbers fluctuate all the time. I've read, it's hard to get exact numbers sometimes when it comes to their bottleneck. So their bottleneck, they oftentimes say, was 70,000 years ago, where the human population was reduced to 2 to 10,000. So sometime before there, probably between 100 to 200,000 years. And the issue was when they supposedly left Africa, migrated out of Africa, they ended up finding other types of humans like Neanderthalensis, Denisovans, for example, Heidelbergensis. You had your hobbits like Floresiensis, Homo luzonensis, Naledi, these ones that were on islands outside of Africa and experience accelerated genetic degeneration, which would explain all these so-called primitive features. So their numbers fluctuate, but that's what's funny is when they're relying on low quality science, indirect science. Okay. And what I like about our guest that we're going to be having on next week is his books talk about that very thing, direct measurements and indirect measurements. The direct measurements, Ramad and I discussed for hours the mutation rates, and we went into detail on each pedigree-based study, the empirical method, and even the ones that give, you can say, okay, you got dates that give 6,500 years all the way up to 12,000, 17,000. Here's what's funny. Nowhere near their date of 100 to 200,000 years, like Shadow Dancer is saying, the storyboards that they pull out to try and get their Eve or their Y chromosome Adam date within that 100 to 200,000 years is priceless. It's hilarious. So yeah, and their bottleneck in the out of Africa. So inbreeding, here's the problem. And they don't really know how to explain our model, because we explain the vast majority of DNA differences as the result of initial creation, front-loaded genetic diversity, okay? At creation, there'd be no mutations. After the flood, this is probably where we started having some accelerated mutations. Of course, after the flood, we'd have genetic drift. Small populations, genetic drift can lead to rapid fixation for that time being, which would explain as well the hypervariable regions. And the evolutionists forget about Babel. So we have all the animal kinds that actually spread out around the world. We have humans that, let's say there was 1,000 that congregate in Babel. Okay. There's another bottleneck where they're all inbreeding. Okay. Which would explain why humans today have low genetic diversity. So not only did God create two humans, Adam and Eve, we'd expect low genetic diversity. You have the flood bottleneck. The animals go and migrate all parts of the world. But then you got the Babel event where, once again, humans congregate. I think there's probably about 1,000 people, maybe a little bit more at that time, between 1,000 and 10,000. They're inbreeding. You got another bottleneck, more reductions in genetic diversity. And today we see that humans have low genetic diversity, incredibly low genetic diversity. This all didn't have to be true, but it is true. It turned out to be true, consistent with the biblical creation model. We haven't had to resort to fancy stories about hypothetical population bottlenecks where the evolutionist explains all DNA differences as a result of what? Genetic mistakes over time, okay? Mutations, whether they want to admit it or not, are akin to typographical errors in a text. You get to that bottleneck in the out of Africa scenario, we've got 2 to 10,000. And I was asking a geophysicist the other day on John Maddox's after show, this same question, and David Neff as well. And this geophysicist said, oh, I've been at this for years, 10 years, 12 years. I said, okay, explain how this out of Africa scenario, according to your model, you're the one that explains the vast majority of DNA differences. Well, no, they explain all DNA differences as a result of mutation. We don't. We explain the majority of nuclear DNA differences as divine creation. That's why inbreeding would not have been a problem at the creation event, of course. Inbreeding reveals the hidden reservoir of genetic mistakes. If there's no genetic mistakes, there's not going to be accelerated genetic degeneration like we see with the cheetahs today, like we see in the evolutionary model in the out of Africa scenario, this 2 to 10,000. It wasn't one generation followed by rapid and exponential population growth like our model. No, this population bottleneck was reduced to between 2 to 10. I've heard people say 30,000, give them that 2 to 30,000 for over 1,000 years, well over 1,000 years, because this would have to be the case. Speed of sound always argues this, but it's like, buddy, it has to be the case because the reason why you're invoking it is to reduce levels of genetic diversity, reduce levels of heterozygosity and increase levels of homozygosity. That's what they need. But at the same time, all of these genetic mistakes that have been accumulating, okay, for millions of years, they would now come to the forefront. They would be manifested. They would lead to rapid, accelerated genetic degeneration. They would become fixated. Fixated means to get stuck, to be stuck in place, fixed. These deleterious mutations would be fixed, leading to genetic degeneration. But then they want us to believe that this highly degenerated population of 2 to 10,000 suddenly exploded into all parts of the world, seizing dominion over the planet. Nothing but fancy storytelling with no real evidence backing it up. The inbreeding backs us up. That explains the so-called hominins, the what they say are the pre-humans. So the numbers have been done, and the numerical simulations have been done. And I'm not saying all Christians and creationists need to go read these highly technical papers about numerical simulations or go watch these lengthy lectures on the HapMap data and allele frequencies and the spectrum of alleles in the human populations and deep, sophisticated population genetic stuff. But it's just so funny that now they know they've been backed up. I showed the comments that they're getting. I mean, these aren't the only ones. I mean, this girl, these people in the comment section, that's Dan's video. They can see that they're backed up in a corner. They're not doing too well. Erica tries to say, oh, my viewers, they know better to come to your channel. No, they come to the channel, they see the videos, and they beg their profits for a response so they can wipe off their sweat. And this is what's going to happen. I'm going to predict someone like Erica is going to come out with a video. She's going to dodge, dip, duck, dive, and dodge. But just because there was she could literally put response video to SFT and raw mat. And the video could be 10 seconds where she just stares into the camera. And they'll think she won because they're just looking for confirmation bias. It's actually kind of hilarious. So for anybody who just joined, we got a good group of people. Be here Thursday. Dr. Dino, I love when Kent's here. He makes for some really entertaining debates. The Devolution shirt is awesome. I'm telling you, I spent all day getting notes, prepping for this show. So if you want all the gear right here, you got to love the mug. Pawns come to people. Like I said, Rob Stadler. This book right here, The Scientific Approach to Evolution, What They Didn't Teach You in Biology. Fascinating book so far. Like I said, he goes over to high confidence science, which is direct science, that which is testable, repeatable. Origin of life, we're never going to get an answer to my challenge. They're never going to answer the questions. I could ask them again here for 15 minutes. Doesn't matter. I've already asked it. Plus, we got the full lecture if they want to go through that one. High confidence science. That's what we're doing. We're looking at the empirical method, empirical mutation rates, and they're getting pretty desperate. So let's see here. I think I had another paper that I wanted to. Yeah, I love this guy right here. Pawns come to people. So, okay. So the whole point is out of Africa it's a fraud. Chris Keith, Do you have that devolution shirt and a long sleeve, right? I think I do, brother. I think we do. I'll put the link in the description box for the website. I got a Ponds Come to People shirt. Really good quality too. Let's see here. Who else we got in the chat? Shadow Dancer, keep up the good fight. Jason, good to see you, brother. Dessel's still here. Shadow Dancer, I got to find where your comment is so I can remove it off the screen. I don't know where. The chat's wild. It's lively. I can't keep up. Yeah, so the next argument we're going to go at is going to be do you have the drooling scientist as a whiskey flask? Maybe we should get that. I don't know if we want to promote the alcohol, though, but that is, I mean, like you were saying, Dessel, he looks like he's on the good stuff. Shadow Dancer, I can't find your question. I can't remove this off the screen. It's going to distract. Okay, we'll keep going up. This is good, though. Lots of good comments. Seems good to that was a good question, Jesse. That took me on a rant about the out of Africa. You know how I love that. Out of Africa is bunk. It is. It is. I've been working hard on a out of Africa book. And, well, see, I've been asking those same, even when I asked Gutsick Gibbon in our first debate, I asked her that same question. How's it remotely feasible? And they always resort. Go watch these debates. They always say, well, you know, this is always the response. Well, I mean, look at the cheetahs today. They're surviving. They experience a bottleneck. Cheetahs are now down to 7,000. They're highly degenerate. They're all like identical twins. Conservationists are worried of their extinction. Are these cheetahs suddenly going to explode into all parts of the world, seizing dominion over the planet? No. You know, when you want to explain all DNA differences as a result of mutations over time, well, then you'd better expect major problems in your bottlenecks that aren't one generation like us, followed by exponential population growth. No, they're 1,000 years. Chris Keith, brother, when I looked at it, I thought I saw it was available in long sleeve. I think it is. Yeah, I think it is. So what I find funny too, just because I saw SFB Mob mention natural selection, these evolutionists, okay, they want to fight all day, but they're so desperate about just giving us one beneficial mutation that supposedly, you know, can counterbalance all of the deleterious mutations that are just pouring into our genetics. Generation after generation, you know, the nuclear DNA, the nuclear genome got about 100 new mutations per person per generation. The best beneficial mutations they can point to are either just organismal adaptation, epigenetic, for example, reductive. I mean, sickle cell anemia, there's a significant impact, but it's reductive. It's due to broken gene, broken protein, broken cell. I mean, these things are not taking things forward. And just for sake of argument, I'll give you 2, I'll give you 3, I'll give you 5. 5 rare, truly beneficial mutations, which probably don't even exist, but let's say they do. It's not going to counterbalance all the damage. While you're trying to improve a couple nucleotide sites, you've got vast numbers of other nucleotide sites degrading, losing information. You're not going to make up for all that information loss by 1 or 2 beneficial mutations. So anyways, that was kind of a rabbit trail. So let's go to the hyper variable argument. We got brother Bill Morgan in the stream. So me and Bill, we've been talking, and Bill's the man. He's one of my favorites. He comes into here, and he puts on a great show, always makes for good debate. So what we're going to be doing is we're going to be doing a biology and genetics made easy, where Bill, myself, hopefully John, maybe Matt, we're going to get in here and we're going to break down that which is really technical and explain it for the layman. Almost, I want to feel like I'm explaining it to my children. You know, when we say created heterozygosity, all that means is God created the original kinds, Adam and Eve, with amazing genomes that were given the ability to adapt, to change, to speciate. That's all that means. So we're going to be doing that series, and I'm looking forward to it. So, Bill, it's good to see you. I think I've seen a question from, if anyone in the chat can let me know, can you see the screen? Dessel asked the question. SFT, how would you respond to the idea of natural rafting for Homo erectus and floresiensis to the Indonesian islands? Neff appeals to mass rafting from Ararat post-flood for biogeography. Yeah, I mean, for the post-flood, land bridges would be, I mean, all of that water would be tossed into the ice caps. You could walk nearly everywhere on the planet. If I could get that slide where it's from, I believe, what is it, an image from NASA or something where it shows if the water was all thrown into the ice caps like that, you can almost walk anywhere. Now, there would be some natural rafting with, you know, there'd be some situations like that to humans, even taking different types of animals to different parts of the world. But that brings to mind, Dessel, because a lot of paleo experts, they'll say that Homo floresiensis, which are the hobbits, they're a degenerate form of erectus. And erectus, for the most part, is human, even according to the evolutionists. Now, they say they're a little bit pre-human. We would beg to differ. But here's the thing. Erectus was known for sailing. It had the ability to sail. So it makes sense that Erectus would have sailed. Purposeful navigation, okay? Screams out intelligence. They would have sailed to the island of Flores, and they would have experienced rapid accelerated genetic degeneration, insular dwarfism. And that's what we see with the hobbits, okay? This makes sense. Erica used to agree with this, but now it's too close for comfort. So she's making all these videos trying to say, no, you know, the hobbits came from the Australopithecine. So she's trying to say an Australopithecine, essentially an ape. Obviously, she would never say that they knew how to navigate and sail. So they would have accidentally somehow, maybe one or two, but an entire breeding population, I don't think so. It's a far-fetched story. It's much more parsimonious to just say and agree with the paleo experts that say Homo erectus sailed purposely to the island, and then insular dwarfism or island dwarfism, it's a known thing. So she's just kind of desperate with this argument because it's too close to comfort. They don't want to agree with anything, because then that would agree with our model that erectus is a degenerate form of humans post-Babel, and the hobbits are a more degenerate form of erectus, which is what the paleo experts say. Now, what Erica will do, this is her trick. She'll pick and choose papers. So she'll pick like a most recent paper of somebody trying to say that, hey, look at these primitive features. You know, they look like they're Australopith-like. So maybe they came from the Australopithecines, but then she'll ignore all the other papers that suggest otherwise when those primitive features can be seen in degenerate humans. You don't have to conclude they're Australopithecines. Here's her little trick. She thinks just because it's the newest paper out there, that it's the best. A lot of times these conclusions that were made in the past and they're overturned for oftentimes it's peer pressure reasons, you know, especially with like Sadiba, where they agreed at one point that some paleo experts concluded that it's an artificial species, and we're going to get into that in a different video. But, you know, one of them supposedly changed their mind. But when you've got the pressure where, you know, we're lacking transitionals, this has to be a transitional, you know, sometimes the original conclusion is the better conclusion, and that's the issue. So I'd like to see how Erica would explain some Australopithecine getting to that island. Sailing, I think that's the toughest question for her to ask. So yeah, so good to see you, Bill. I'm looking forward to that series that we're going to be working on. Okay, so screen should be shared. So Dan made okay, good, here it is. So hopefully everybody can see. So Dan made a video 2 weeks ago about supposedly the hypervariable regions, and then failed to address my argument, my comment. I'll show you what his response was to this, and just went on using the same argument like it hasn't been addressed. So here, what I say is, all God has to do is engineer 10 million variants in Adam and Eve, and everything else is explained. Well, You'll see that even in the HapMap data, there's about between 10 and 15 million common variants. Okay, so those would have been the ones that were in Adam. Those are common, found all over the world. So they would have had to have been present at Babel, present at the flood, present at creation in Adam and Eve. Now, what I say here is this actually makes sense considering there are between 10, exactly what I just said, 10 and 15 million common variants worldwide. We actually, here's a good point. We actually have roughly 3 million, okay, individually. Adam would have 10 million, not difficult. Original created genotypes is entirely plausible given the absolute failure of abiogenesis and origins of life research. Once again, the ones that want to scoff at the front-loaded model, I mean, that's our assumption, makes testable predictions. As I showed earlier, it's the numerical simulations bear it out. Definitely looking forward to responses on that. They better answer my challenge on abiogenesis because so far they're failing miserably. So anyways, most of the world's variation is 2 alleles, A or C, G or T, with an allele being a flavor of a gene. After creation, we have thousands of years for mutations to build up. Therefore, many alleles, ones restricted to certain populations, are the result of these mutations over time. For example, blue eyes, okay? I point out to Dr. Dan, there are some exceptions to the rule, which he doesn't really like to. This is what they do. They like to point to the exception to the rule, to overrule the rule. This doesn't make any sense. So yeah, and he talks about this a lot in his newest video. So we have some cases where we have several hundred alleles or variants. One example is the human immune system, okay? There is an extensive amount of variation in the immune system among people worldwide. We have 7 billion people or more today. Think about how many family trees there are. You start with Adam and you begin to branch outwards, okay? Especially because so many parts of our genome are more prone to mutations. That's why it looks like a lot of these changes are due to pre-existing algorithms, responses from the environment, epigenetic factors. We have millions of DNA switches, millions and millions in our genetics, just waiting to be turned on. Many genes, like many non-coding RNA genes, such as pseudogenes, they're only expressed under certain conditions. I love when they point to the genetic knockout test in mice. You could have gene A, gene B. You knock out gene A and everything's fine. You knock out gene B, everything's fine. You knock out gene A and B, and suddenly it's lethal because there's so much interconnectedness going on. And their arguments are just getting weaker and weaker. So let's see. We have plenty of time for a mutation to happen in multiple places. Given this fact, with highly mutable genes that we can survive with, it turns out we have lots of variation. It also looks like we can inherit a lot of traits that we acquire while we are alive. Actually, Carter put out a good video today on epigenetics where he talks about that. I definitely recommend it. These hypermutable genes used in our immune system, meaning it would be highly profitable to have lots of immune system variants. It is therefore within the design parameters of this class of genes, as you can see here, to be highly mutable with intention. So no, there isn't a problem with created heterozygosity. That claim would be an evolution myth, blah, 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 blah. So his response, standing for truth, sounds like a whole lot of ad hoc, post hoc rescue devices. When in fact, it's just us looking at the hypervariable regions in the genome in people groups in the planet, looking at common variants, looking at allele frequencies, looking at allele frequencies, alleles that are rare. These are just all observations. A rule of thumb when people say, how do you know what's created, what's not? Well, the more common it is, and if it's not disease causing, genetic entropy could lead to disease. But for the most part, if it's not disease causing, it's functional, it's created. The ones that are rare, those would be mutations over time. Yeah, so he doesn't give an answer there. I think I have a couple slides on it too. That was kind of me explaining it, but I mean, you can get really, really technical here. I'm going to leave this slide up because Dan actually talks about some human genes that have over 800 variants, maybe 1,000. So here's a slide on here that I'm going to go over too, just to leave no stone unturned. I understand tonight's a technical night, but it's kind of necessary. So I'm going to run upstairs for 2 minutes to get another bottle of water. And not sure if you guys can see that on the screen, but I'll be back to go over it. Just a second. Okay, I'm back. Maybe someone in the chat let me know that they can hear me. I want to make sure that I know SpongeBob Imagination. I think that's the first time I've ever had dead air. But that's what I get for rushing into this stream with also trying to fight a cold at the same time. But hey, I made it an hour. And usually I got somebody here with me. So it's like, hey, Matt, take over while I run upstairs to pretend I'm going to get a bottle of water. But really, I'm going to take a whiz like Mitchell said, put the seat down. I'd better. I've got a wife. Okay, so might be difficult to see, but here we go. We're going to get a little more technical here. Leave no stone unturned. The comment I left was kind of just given the basics. So here we go. Some human genes have over 800 variants. This is what Dan is saying. Now, these are the exception to the rule, and you are probably referring to a subset of the immune system genes that are designed to change rapidly. Most common variants actually come in two versions. Real quick, check the chat. Make sure everybody can hear me okay. Okay, so I'm guessing the thumbs up. Everybody can hear me okay? Okay, so an exception might be the ABO blood alleles. But the most common, I'll keep looking at the chat just to make sure, but the most common version of O is clearly a mutated version of A. So it looks like there were two originals, A and B, and you can read a whole article on it. O must have entered the human population early. Thanks, Shadow Dancer. Definitely pre-Babel and possibly pre-flood. So yes, Adam and Eve could not carry more than 4 alleles per locus. Among them, assuming God did not pack multiple different genomes in Adam and Eve's reproductive cells, which could have been the case. We can't really limit God. The only thing is the 10 to 15 million common variants is all we would need to explain what we need to explain. But the bulk of the genetic variation discovered to date could be fit into Adam alone. Thus, the prior assumption seems valid. Add post-fall mutations to the mix, and I believe we have a good explanation of human genetic history and genesis. Number of reasons here. So this is Dr. Carter. Must-read paper is can mutations add new information? So he says here, yes, most mutations are bad. But why would the current human population be limited to the number of alleles per locus that were on the ark? Mutations since the flood have added to the diversity. Please note, I take exception to the statement that creationists believe no new genetic information can appear through mutation. In fact, I have submitted an article to the Journal of Creation on the subject that will hopefully make it through. Let's see. So he says, I believe mutation can corrupt information, scramble information, and change it horizontally. So his point would be, although it can change, it can adapt, which in a sense could be, depending on how you want to define new, could be new. But the thing is, it's the change is down. It's never vertical. So he continues, but that is utterly unable to account for the changes necessary for long-term evolution. Also, evidence is accumulating for the existence, as I was talking about this earlier, of genetic algorithms designed to facilitate changes to the DNA sequence. Actually, this is a term I use frequently too. I really like it. There's something called variation-inducing genetic elements, okay? And we happily incorporate them into the creation model. So what they're referring to there on the variation-inducing genetic elements is like your endogenous retroviruses, your ALUs, your other subclasses of retrotransposons. Given the fact that we now know they're highly functional and important to the genome, they would have also been front-loaded. And Brother Chris Keith, you're the man. I appreciate the support. I noticed you asked a question earlier. Front-loaded equals created heterozygosity. You're right. Yeah. Front-loaded genetic diversity, created heterozygosity, essentially the same thing. So to continue, since these changes would be caused by pre-programmed genetic modules, it is hard to call these changes mutations. Okay, so this is important here, and this is what the data suggests. Actually, you know, I'd love to see Dan fight some of this data where this really it would be the most parsimonious conclusion. So, the HLA genes in particular, as a vital part of the immune system, are pre-programmed to scramble. So using them as proof for your beliefs is an error on your part. The HLA genes. Let's see here. Given that some places in the genome do have high diversity, once again, that's exactly, I'll highlight it. That's exactly what Dan's saying as an argument. He spends a lot of the video on this one. So this is what this is it's some of the stuff I said, but it's a response from Dr. Carter to a critic in the creation website that used the same argument as Dr. Dan. So these are not new. They've been dealt with, and he's dealing with it here. So Dr. Carter says, you still fail to account for the distribution of those variations worldwide. This is very important, guys. In fact, most variations come in 2 main flavors. Adam and Eve could have given us up to 4. And these are generally found in all populations. When we find more variation than this, almost always the 3rd, 4th, 5th, etc. versions are restricted to one subpopulation or another. These are mutations that have occurred since the Tower of Babel. And the exceptions, goes over a number of those. We talked about this earlier, but I'll go over it again. The ABO blood type locus, 2 with 3 main alleles found across the world, one of which, being O, is a broken version of another, being A, could have been due to mutations between Adam and Babel. Interestingly, deletion mutations, which are common within all people, are generally not shared among the various subpopulations. This is evidence of a young genome in rapid decay. I wonder if they'll actually address the bigger picture, the lines of evidence from the HapMap data, for example, that really do suggest a young genome. I'm not too sure if those will be addressed. Dr. Jeanson actually has an awesome lecture on this too from the last couple months. Adam and Eve, going into population genetics and touching on the fact that small populations, according to, because our biblical flood model, that's 8 people, 3 reproducing couples. Okay, you're going to get a lot of mutations that can be fixated quickly within those generations, and could explain a lot of the hypervariable regions we see today. Now, I wanted to kind of put something in a nutshell. You're going to find this in Matt and I's new mutation rate book, but to kind of put it into a nutshell, fixation rates, and like I said earlier, when you hear the word fixation, all it means is to get stuck, to be stuck in place, to be fixated, essentially. Now, they occur at fairly consistent rates in all humans, okay? But why different people groups have different amounts of fixation is because some people groups remained in small populations over time, and smaller populations then reach fixation faster than larger ones. This is why most of the diversity is in Africa. They have small tribes, and they have larger civilizations. So, and Dan also in his video talked about the inbreeding, the Neanderthal genomes, or how inbreeding doesn't result in faster mutation rates, but we've addressed all that ad nauseum. So I'm going to link all the other videos that haven't been responded to. So instead of going over the same thing again for an hour, I'll link those for you guys to check out. Here, see a source, all human beings are 99.9% identical. Didn't have to be true. The 1Y chromosomal line didn't have to be true. 1 mitochondrial DNA line didn't have to be true. Didn't have to be true for evolution to be true, but had to be true for the Bible to be true. And yet it was true. How did the evolutionists explain that? Paper here, harmful protein coding mutations in people arose largely in the past 5,000 years. Another one, gene mutations began showing up in the last 5,000 years of human evolution. What a coincidence. Still waiting for some arguments on the confirmed predictions by Jeanson on Y chromosome. So a ton of good stuff here. Another great one, 80% of proteins are different between humans and chimps. This one, Matt and I talked about in great detail, the Eve mitochondrial consensus sequence. Great paper. We know Eve's sequence, and I explained how we could figure it out, how we could know it. And that was probably maybe 6 videos ago in our Refuting the Critics series. There hasn't been any response to it, so I definitely recommend going to watch that. Yes, I mean, there's just a ton of great science that demonstrates the validity of the biblical model. So let's see how our wonderful chat's doing. Yeah, I'm definitely looking. Mitchell says, I wish my genome had high diversity. Don't worry, none of our genomes got high diversity. Shadow Dancer, she says, is there a paper that shows mutations that bring new information? It's a really good paper from Dr. Carter. So it's not new novel information being added. What he's saying is that there can be adaptive changes in our genome that can lead to an advantage, right? But still, they're typically at the expense of reduction. They're typically functionally compromising to the organism. But in a way, depending on how you define new, a new function that's popped up, or a new benefit that's popped up through a reduction, or even just through an epigenetic change, right? Where it's just general organismal adaptation because that's front loaded and built in. One could say that's new, but not new in the sense, these would be horizontal changes is the best way to look at it, okay? Deep time, large scale evolution would need that which is vertical changes, and that we don't see. But yeah, I'll send that to you. Awesome. Yeah, let me check my notes, see if there's anything else. I'm definitely getting kind of tired, but leave no stone unturned with these guys. Once again, today's Tuesday, so Thursday we're going to have Kent, and looking forward to hopefully getting Carter on, Dr. Carter. Actually, one other thing I want to point out is, so I address this, how they underestimate the diversity with how they define allele. I'll end it with this. In my book, Refuting the Critics, I go over how Dr. Jeanson and Dr. Frello have had an extensive written debate. Now here's, I don't believe Dan, and I don't believe these critics when they say, oh, they don't have the time, or they don't want to publish their objections in the Answers in Genesis journal because it's not a reputable journal. Nothing but excuses. If they have all of this time to make their videos, some of them will spend all day in the comment section. Like our good friend CRISPR, he'll spend the whole day I limit my time in the comment sections or I'll lose my mind. They're constantly in these blog websites. Why don't you spend one day, give a 10 to 20 page rebuttal to Dr. Jeanson, put it in the AIG journal, and then Jeanson will respond. So yeah, Jeanson is not scared one bit of these guys. So I addressed this. This was actually, and then I referenced the debate with Frello and Jeanson. So I'm going to read it here from my book here. So Frello has chosen to utilize this poor argument against the created heterozygosity hypothesis. Now, Jeanson says this, okay? Jeanson says, let's start by identifying where Frello's claims and representations of replacing Darwin are correct. He goes over a number of things that Frello's correct in. But then Jeanson points out, and this is something Dan and Swamidas and all these guys are guilty of too. And this is why they don't want to do what Dr. Frello did, okay? Because Dr. Frello uses a lot of the same arguments as these guys. He's a PhD. He knows what he's doing. I've debated him. And we're still waiting on Frello's response to Jenson's last rebuttal, and there's been none. But these guys, they know once they agree to accountability and take Jeanson on in this format, they're going to get exposed the way Frello did. So Jeanson points out, but then Frello's argument commits a common genetic mistake. In his endnote, he correctly defines an allele as one of two or more versions of a particular DNA position. But then he contradicts himself in the paragraph above. These guys are always contradicting themselves. A maximum of 4 alleles, Frello says, of each gene could be present in this original pair. So he's talking about pairs on the arc, if you read the whole written debate. A maximum of 4 alleles of each gene could be present in this original pair, Frello says. By definition, Jeanson says, genes consist of more than one DNA position. So which is it, Frello? Is an allele a version of a particular DNA position or a version of a gene that contains hundreds to thousands of DNA positions. Frello can't have it both ways. See, these evolutionists oftentimes want it both ways. I'll move on down the page. Jeanson continues. In fact, his argument rests on his erroneous adoption of the second definition, that alleles are different versions of genes. Under this definition, he misrepresents my explanation, this is Jeanson talking, for nuclear DNA differences. In fact, in one of my published papers that I refer to at least 15 times in Replacing Darwin, I explicitly addressed Frello's error. And this is one of the papers, actually, that Dan referenced in his video the other day. So I guess he missed it too. He says, if an allele is defined in terms of a gene unit, then generating allelic diversity by mutating just one gene per mutational event produces little diversity. Instead, if an allele is defined as a single genomic position, independent of its relationship to a gene, then enormous allelic diversity can be generated by mutation. This is still Jeanson talking. As an aside, allelic diversity need not arise via mutation. Again, if we use the genomic position definition of an allele rather than the gene unit definition, other mechanisms besides mutation can generate allelic diversity. He gives a really good example here. So he says, for example, a single gene typically spans thousands of nucleotides, and SNVs might be distributed throughout the gene, for example, at 90 of the nucleotides within the gene. If we allow for the genomic position definition of alleles, every single one of these 90 SNVs may have existed in a heterozygous state in each of the individuals of the pairs brought on board the ark. And then this is important. So Jeanson says, expanding this single gene example across the entire genome reveals a tremendous potential for allelic diversity on the ark. In just 2 diploid individuals, 4 genome copies exist. Since only 4 DNA base pairs exist, virtually every possible genomic position allele, which is far more than 4 to 28 gene unit alleles, could have been present at the time of the flood if the individuals were heterozygous. And then here's the thing, though. In the next written part, when Frello comes back, he essentially agrees that Jeanson corrected him. So what's funny is the fact that somebody like Dan, he can spend all day in blogs, comment sections, making videos. Go do what Frello did. Put in your rebuttal. Guys, thanks so much. Super sticker, another super sticker, super chats. You guys make this worthwhile. We want to put out as much content as we can. Like I said, Bill Morgan and I, we're going to hopefully soon start working on the biology and genetics made easy. Okay, I'm gonna share screen. That's an hour and a half of demolishing damage control Dan and Team Dodgeball. What's funny is I would challenge Erica to just without any help go through this video and debunk it, since she's so outspoken, but she won't. You know, she's going to have to go to Evograd or go to, which is fine. You can't be masters and everything, but these people are so militant, you know, that young earth creation is apparently wrong. But this one will have to be for Dan to deal with, but it's not going to be possible. So I hope I got it up here. Let me see. Oh, here it is. Okay. This is funny, guys. So hopefully you can see. Creationists invent their own mutation rate. This was in 2016. Now this, if you read through this, this is where the ripple effect started with these guys. Dan, Evograd, Herman Mays, and then your copiers. Well, I guess they're all copiers, but your really, really bad copiers like your Erica. This is where it all started. I'm going to check from my phone, make sure you can see the site. Okay, good. Mitchell, Doki Doki, thanks so much. Good, you can see it. So this is funny. All right. So they talk about how Dr. Nathaniel Jeanson published a study on molecular clocks, blah, 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 blah. So if you read through this, it's all your same typical arguments, you know, the same ones that Mays uses, same ones that Evograd uses, same ones that Dan uses. Here's what's funny, okay? Here's the same thing that I showed you earlier where that Stacy girl was admitting that the creationists are winning, begging these guys to step up their game. Like I said the other day, somebody was begging Dan to talk to Erica to respond to our videos. You know, these guys, and here's the thing, they're always welcome in the spiritual family. We want them to be converted. Chances of the militant ones being converted are slim, but it's the ones that are open-minded, on the fence. Shadow Dancer, thanks so much. I appreciate that. Okay, so you go down. Guess who it is? So hopefully you guys can see this. Dr. Jeanson's reply to this article. Now, I started getting tired personally of responding to the same arguments over and over again, okay? So I took this response and started posting it in the comment sections and said, deal with this. This is all the same arguments Dan uses. It's been dealt with. And nobody can refute it. So Jeanson goes hardcore here. Jeanson says the main claim that you can find this in the comment section of this blog, because they'll say, oh, you know, Jeanson hasn't dealt. Oh, he's dealt with it, but he hasn't had any response in years. The main claim is that my mutation rate is 35 times faster than the published one. And he goes over the fact that it's circular reasoning, the way they do it. Let's see. Jeanson covers the heteroplasmy to homoplasmy. Right here, he corrects them about, because here's the thing, there's a double standard. So Jeanson here says, the author of the blog took for granted that number of mitochondrial DNA differences among the different people groups. But how many of these have been validated as germline changes and not somatic ones? For example, the author has no problem with my citation of 123 differences being the max difference between two humans. But how does the blog author know that these differences are germline ones? And what's funny is I was talking to somebody the other day privately. I won't say who their name was, but they're oftentimes referenced in these. And they even agreed, although they play semantics all the time, maybe suddenly they'll disagree now, I don't know. But a lot of these people, they're not even worried about this whole dyad, triad thing, somatic versus germline. We can know, we can be confident if it's germline, okay? It's a silly argument. I said earlier, it's been done on autosomes, and there's very little difference between the two. So Jeanson continues, the evolutionary mitochondrial Eve and out of Africa model. Caleb, Doki Doki, thanks so much, guys. I appreciate that. That gives me the energy I need to keep going. So, guys, you got to read this. This is awesome. I'll show you the funny part in a second. Okay, so here's a good one. Jeanson continues. So he destroys this, okay? He says, if the rate I cited from Ding's data is invalid, why does it agree with the 7+ studies that have been previously published? Why is there such strong scientific consistency across multiple independent scientific studies? If the blog author wishes to question the conclusions of the one study he cites, he has a lot more evaluating and explaining to do than the single paper which he interacts. So that being said, we put out so many videos recently where Matt and I just went through every single pedigree-based study, went through all the numbers, and Dan, because he's tapped out. He has tapped out. It's so obvious. I mean, if this is their best, it's sad. So, and he'll never admit it, but it's just so obvious. So Team Dodgeball, they say that, oh, we don't want to hear, you know, raw, mad, and standing for truth go over all these pedigree studies. Yeah, because you can't address it. You can't refute it. It's contradictory to what you've been saying, to your model. Redefine living. Thanks, brother. I appreciate it. Okay, so Jeanson continues. In the fruit fly, roundworm, water flea, and yeast mitochondrial mutation rate studies, the rates were measured in mutation accumulation lines. In other words, over several generations of genealogically related individuals. And here's the thing, most evolutionists, like, you know, we got Dessel Drayson, who I appreciate. He's objective in all this. He likes debates, and he watches them objectively. I bet he doesn't know that Jeanson responded to these guys, and they never responded back. Because they want to hide. This is the evolution destruction that they don't want you to know about. And of course, here we go. Okay, so he continues. Almost by definition, these studies measure germline mutations, not somatic ones. Why do all these studies agree so strongly? Another answer that we'll never get. What testable prediction does the blog author's model make? Months and months, they've all failed to provide any. Then damage control Dan comes in, provides one. Me and Matt tear it apart, showing that it's not novel. It's not future novel testable, so they have none. Jeanson points out this is the gold standard of science, the one to which creationists have been held to for years. Watch this. What was this, 5 years ago, 4 years ago this was made? Jeanson says, if the blog author thinks that he has a better answer than what I have published, I challenge him to make, look, here we go again. I challenge him to make scientifically testable predictions from it. And look at this, right from Jeanson himself. For example, from my young Earth creation model, I can predict, in brackets, he says, I'm claiming that I can. He's taking the challenge to him. I've been taking the challenge to him. We all have. The mitochondrial mutation rate in the millions of species in which it is yet to be measured. I'm willing to test my predictions in the lab. He's also willing to debate any of these guys if they want to be held accountable. I even told Dan as a joke I'd pay him. If it was a reasonable amount, I probably would pay him to do it just to watch him get demolished. But the thing is, we already are demolishing them, so what's the point? Okay, so he says, I challenge once again the blog author to do the same. Otherwise, by the evolutionist's own standards, the blog author's claims are pseudoscience. Okay, Herman Mays, look at this. So 2018, okay, so no, the blog author didn't respond, didn't take up the challenge, and Jeanson 2017. So 2018, nobody comments, everybody's silent. Good old Herman Mays, look what he says. I realize the post is old and it's been ignored. And this is the great Herman Mays, right? And even the last comment is over a year old. And look what he says here. But I would like to see your response to Jenson's rebuttal. For the record, I think it is absurd. Strawman's Jeanson doesn't really give anything substantial here. Look at the next comment. You're not the first one to ask this. Notice that? You're not the first one to ask this. Always looking for damage control. Always looking for that next prophet of science fiction-based religion, pawns come to people evolution, to come in and save the day. Nobody saved the day yet. And look at this. I'm also curious about a possible response. Look at him here. So I have been trying to get in touch with the author. Looks like the author left the country once he realized his entire article just got schooled. Oh, and then he says this. He's been trying to get a hold of the author. Sadly, not much luck. Let's see. Yeah, so, yeah, and then it ends. So never any response, because they don't have one. And I went into Reddit, and I don't want to find it now. You can look for it, take my word for it, unless it's been taken down. I saw in Reddit, I believe it was one of Dan's posts or one of the ones he was involved in. And there was somebody in there who was saying, can somebody please address Jenson's comment? And still nothing. So guys, this is what we're dealing with. Dealing with people who bring up the same arguments, professional dodgeball players, not answering the bigger questions. George Bond, thanks so much for the super. Brother Ramat says, they know they will get exposed and it will be made public. Exactly. It'll be made public. Like, for example, the Frello and Jeanson debate is Frello's now being held accountable. They both are. It's out there to the public. It's published. They don't want Evograd, he's not going to. I would debate Evograd. I don't think he would, but I would, since he's the one they all look to. So Matt says it'll be made public by AIG and they will never live it down. That is the real reason they do not debate. Tell you what, every single subscriber of mine will pitch in $1 for Dan to do it. Let's see what his excuse is now. He thinks we actually buy this nonsense. All we can do is laugh, though. So, Matt, you're totally right there, brother. Doki Doki, thanks so much. Yeah, that Vice Rhino video was great. Taking on these big dogs is going to be fun. So let's see here. Got anything else? There he is. Evolution is a fact. Pond scum to people. Well, unless there's, I think we've tackled that pretty good. I didn't think I can get through all that, but it's been an hour and 40 minutes. We did. Once again, challenges out there to these guys. And as Doki Doki says, spoiler alert, Dan, the pseudoscience man, is going to play dodgeball. Yep, they've mastered it. The 5 Ds of dodge. And Shadow Dancer says, Dessel Trace, my picture. Dessel, we'll send you one of the Pawn Scum to People shirts. I got a workout shirt and the Pawn Scum to People one. So Professor Stick, yeah, we're going to be taking him on soon, Jesse. I'm excited for that one. See, these bigger channels are even easier. Like that Rhino one was just a walk in the park. Professor Stick will be too. Thank you for the super sticker. Okay, so I think I answered all the questions in the chat. Sorry that I wasn't as interactive for a little bit of time there going through those slides. But yeah, it can be technical, you know, the numerical simulations, the HapMap data. Once I'm done here, I'm going to add the necessary sources into the description box for those that want to dig a little deeper. But the point is, they're resorting to sophisticated sounding population genetics and this whole, you know, TMR4A that Dan, he's been at this for years and he just heard about it last week and he throws it into his video and we just demolished it there. Nicholas, I'm Canadian, eh? That's why we love our Tim Hortons. Nick, I'm Canadian. Nicholas, brother, when this is all done, I'm going to invite you over to Canada. We'll hang out. We'll fellowship. Nicholas is awesome, guys. If you haven't yet, please go subscribe to Nicholas's channel. He puts out some great stuff. True brother in Christ. Go subscribe to Shadow Dancer, Raw Matt, Bill Morgan, everybody. Doki Doki, you're awesome. Thank you for the support. We're getting closer and closer to doing this full time, and these guys really won't have anything to say in return. I mean, the science is on our side. So Canadians love syrup and beavers. Yeah, my wife made some pancakes this morning, and I did put lots of syrup on it. So, well, you guys are too much fun. Still got over 30 people in the chat. Could probably do this all night. You know, we'll do one more showing of this Thursday. Dr. Ken Hoven, Derek Barnes. We got to get Bill Morgan on again. He always puts on a good show. Okay, guys, I think that's it. Another irrefutable stream. Amen, Mitchell. Good to see everybody. Thanks for the super stickers, super chats. We love you here at Standing for Truth. Dessel, thanks so much. I appreciate it. If you think I didn't address anything or missed it, comment, email me. For the people that especially are open-minded, even the critics, I love answering the questions. It's only your 0.1%, the militant ones, like your Dr. Dan, Erica, where they ask a question, you give them an answer, and they say you didn't answer it. It's psychological warfare, okay? You know, Dan would just go into the comment section, Dr. Dan, and say, oh, thanks for addressing nothing. When he knows he's been destroyed. He knows his question's been answered. His objections have been overcome. It's a psychological game. So people who aren't aware, or maybe they're not up to date on this, they'll go into the comment section, they'll see that, and they'll think, oh, you know, I don't need to watch this. He didn't address anything. It's all psychological warfare, and it's not going to work on us. So, okay, guys, God bless. And Standing for Truth is out.
And for those who enjoy this channel and would love to support us financially, please feel free to hit that donate link. We'd greatly appreciate it.